Good afternoon. My name is Carina Hicks, and I am with the Department of Psychology at the University of Memphis. Today, I'll be presenting my work in progress titled Dopamine Dynamics in an Animal Model of Attention Deficit Hyperactivity Disorder, the SHR rat. Attention Deficit Hyperactivity Disorder, shortened ADHD, is characterized by symptoms such as inattention, hyperactivity, and compulsive behavior. The Spontaneously Hyperactive Rat, SHR, has been thoroughly researched and has been proven to be an excellent animal model of ADHD. We plan to use in vivo fixed potential amperometry, which will be described in the methods section, to measure and quantify dopamine release, that is, the magnitude of dopamine response, and half-life, the time that dopamine is in the synapse. We will do this in this well-established model to create a baseline for comparison to other lesser-known animal models to look for convergent mechanisms in future projects. For this project, we will use both the SHR rat and the Wistar-Kyoto WKY control as subjects. There will be 48 total animals used, 24 SHR, broken down into 12 male and 12 female, and 24 WKY, also with 12 males and 12 females. Before becoming available for in vivo fixed potential amperometry, rats will undergo operant behavioral testing in a separate lab. In vivo fixed potential amperometry with carbon microfiber electrodes has been established as a valid technique for the monitoring of stimulation-evoked dopamine release of the medial prefrontal cortex in real time. To accomplish this, rats are anesthetized using 2 urethane IP injections and then placed in the surgical stereotaxy setup on a heating pad to maintain temperature. Here, with the help of stereotaxic carriers, we place a carbon microfiber recording electrode in the medial prefrontal cortex, a stimulating electrode in the medial forebrain bundle, and a reference electrode on the surface of the parietal cortex contralateral to the stimulating electrode. After positioning the electrodes, a fixed 0.8 volt current is applied continuously to the recording electrode via an electrometer. This allows us to ensure that we are measuring dopamine, which is known to oxidize at 0.6 volts. Changes in dopamine release and half-life will be monitored continuously from the medial prefrontal cortex in response to chains of short electrical stimulations applied in the medial forebrain bundle as a baseline recording. These stimulations will be 20 pulses at 100 Hz every 30 seconds for 20 minutes. At approximately 20 minutes, all rats receive an acute IP injection of nomifensine in order to assess the percent change in dopamine release and half-life following the use of the drug. After the administration of nomifensine, dopamine oxidation currents are monitored for 1 more hour, followed by continuous stimulation to depletion to quantify total dopamine. Following the recordings, a direct anodic current of 100 milliamps will be applied for 10 seconds to mark electrode positions. Here, you can see an example of what that would look like with the stimulating electrode in the medial forebrain bundle. Rats are then euthanized using an intracardiac lethal dose of urethane. Brains will be removed and stored in a sucrose solution. Coronal sectioning of each brain will occur using a cyrostat at negative 30 degrees Celsius. Electrode placements are then identified using a light microscope and recorded on coronal diagrams. Microfiber recording electrodes are calibrated following use in the experiment. This occurs by exposing the electrode to a series of known dopamine solutions, 0.2 micromolars, 0.4 micromolars, 0.8 micromolars, and 1.2 micromolars via a flow injection system. An ideal example of responses is shown here. We do this to allow us to be able to convert raw data, which is in nanoamps, to dopamine concentration in micromolars. Shown here is response we expect to see after each short stimulation, with dopamine spiking just after the initial stimulation before tapering off again. We also see an example of data of the effects of nomifensine on percent change in half-life. This data comes from the following article that was published from Dr. Lester's lab. The independent variables for this experiment are the strain, SHR and WKY, and also sex. The dependent variables are dopamine release and half-life before nomifensine injection and dopamine release and half-life after nomifensine injection. 4 two-way ANOVA tests will be used to analyze the data. The first will analyze the relationships between sex and strain on baseline dopamine release. The second will analyze the relationships between sex and strain on baseline dopamine half-life. And then the final two will analyze relationships between sex and strain on percent change in dopamine release and then percent change in dopamine half-life after nomifensine injections. Ultimately, we expect that SHR rats will show a hypodopaminergic profile in amperometry. That is, that they will show reduced levels of dopamine as opposed to the WKY control. Because of this, we also expect to see a greater efficacy of nomifensine, which again is a dopamine agonist that works by blocking reuptake of dopamine and raising extracellular amounts of dopamine. To conclude my presentation today, I would like to thank my mentors, Dr. Duranda Lester and Dr. Helen Sable, for allowing me to work on this wonderful project. I would also like to thank graduate students Nick Page and Joshua Potter for being the first to train me and for their hard work, as well as the other undergraduate research assistants in both Dr. Lester and Dr. Sable's labs. And finally, thank you again to Dr. Melinda Jones and the Helen Hardin Honors College for your continued support of not only me and my project, but all students pursuing research.